is going to benefit your university exams as well as your NEET PG entrance and FMG examinations. So for today's class, we'll be talking about heme proteins, their functions, the most important, that is the reactions of heme synthesis in detail. Then we'll be talking about the porphyrias, different types of porphyrias and their clinical manifestations, followed by the degradation of heme, that is the synthesis of biliverdin, then how that biliverdin is converted into other products and finally excreted from the body. In short question and answers, we'll be taking clinical disorders associated like the Reuben-Johnson syndrome, sickle cell anemia, Gilbert-Najar syndrome. Okay. So introducing you to the core features of Oracle. Here at Oracle, we have live classes to elevate your learning experience. Then we also provide you with recorded lectures so that you're able to access them anytime as per your convenience. And apart from that, we also have notes specially curated based on the latest CBME syllabus. Then we also have question banks which have plethora of MCQs so that you're able to assess your knowledge. In here at Oracle, we have three different subscription plans, beginning from the basic, which is for 3 months and comes at 15,000, followed by the standard plan, which is for 6 months and comes for 30,000, and then the mentorship plan, which is for 12 months and comes for 50,000. In the mentorship plan, you'll also be having personalized guidance as well as notes will also be provided. Okay, so with this, let's begin with today's class. Okay, so talking about heme synthesis. Firstly, what is heme? So heme is if I give you one statement, it is a ferrous protoporphyrin. It is a ferrous protoporphyrin. Okay, or you can also say ferroprotoporphyrin. Okay, now I'm going to expand this. Heme is a ferrous protoporphyrin or ferroprotoporphyrin. Now, what do you mean by that? Your heme, it is composed of a metal. So it is having a metal part and it is having porphyrin. So this metal here in heme, this is your iron, and iron is present in the Fe2 plus state, that is the ferrous state. Okay, and you have porphyrin. Now, what is porphyrin? This porphyrin, it is 4 pyrrole rings joined by methynyl bridges. Okay, so I am saying that heme is composed of a metal part, which is your iron in the Fe2 plus state, and porphyrin. And what is porphyrin? Porphyrin, I am saying that you have 4 pyrrole rings in porphyrin, which are joined by methynyl bridges. Okay, now what is pyrrole? So your pyrrole is nothing but pyrrole. It is a 5-membered ring, 5-membered ring with nitrogen. So if I draw a pyrrole for you, it is going to look like this. Okay, so this is a pyrrole. Now I'm saying porphyrin is having 4 such pyrroles. Okay, so your porphyrin, it is a tetrapyrrole. So 4 such pyrrole rings, they are joined together by methynyl bridges. What is methynyl? It is your CH double bond and single bond. So on one side you have double bond and on the other side you have single bond. Okay, these are your methynyl bridges. Okay, so 4 pyrrole rings joined by these methynyl bridges, they form your porphyrin. Okay, so if I draw a porphyrin, porphyrin is going to look like this. 1 pyrrole ring, then you have another pyrrole ring, then you have another pyrrole ring, and then one more pyrrole ring. So you have 1, 2, 3, and 4. 4 pyrrole rings, and these pyrrole rings, as I'm saying, they're joined by methyl bridges. So this and this, and this. So this here, this is going to be your porphyrin. Okay. So this is your porphyrin. It is a tetrapyrrole. That is 4 pyrrole rings joined together by methylene bridges. Okay. And to the center of this porphyrin ring, you have a heme chelated. Okay. In the Fe2 plus state, and that becomes your heme. So this porphyrin, this is a cyclic compound that readily binds metal ions. Okay. Especially your Fe2 plus and Fe3 plus. Okay. And the most prevalent metalloporphyrin in humans is your heme. Okay. So to the center of this protoporphyrin ring, an iron is coordinated to the center of this, and you end up with heme. Okay. So now you know the structure of heme. What is heme? It is a ferroprotoporphyrin. So if you only remember this, that heme is a ferroprotoporphyrin, then you can elaborate on that. That ferro, that it is having a metal part, which is the iron in the Fe2 plus state, and it has a porphyrin. What is porphyrin? Porphyrin is nothing but 4 pyrrole rings joined together by methyl bridges. Okay. So it is a tetrapyrrole. And what is pyrrole? It is simply a 5-membered ring with nitrogen. Okay. So 4 pyrrole joined together by methylene bridges, they form your porphyrin, and this porphyrin to the center of this, when an iron atom in the Fe2 plus state is chelated, or to the center of this, an iron atom is coordinated, you end up with heme. Okay, so heme is the most prevalent metalloporphyrin in humans. Okay, so that was about the structure of heme. Now, another thing, before moving forward, that your porphyrins, this porphyrin ring that I have described, your porphyrins, they are of 3 types. Okay. So this porphyrin ring, that is tetrapyrrole ring, it is of 3 types based on side chains attached to the carbons. That is, you have this pyrrole rings. Okay, 4 such pyrrole rings are there in the structure of porphyrin. So to these carbons, okay, these carbons, they are different side chains attached to these carbons, and depending upon the different side chains that are attached, you have 3 different types of porphyrins. So firstly, I'm going to tell you about the 3 different possible side chains that can be attached to these carbons of the pyrrole ring. What are they? So the possible side chains, what are the possible side chains that can be attached? I'm going to represent them with MPVA. Now, what is MPVA? This is a mnemonic you can remember. M for methyl. So you can have a methyl side chain. P for propanyl. You can have a propanyl side chain. V for vinyl. And A for acetyl. So you can have these 4 possible side chains. Okay. Now, what is methyl? Your methyl is CH3. What is propanyl? It is CH2, CH2, COO minus. And what is vinyl? It is CH double bond CH2, and acetyl is your CH2COO. Okay. So these are the possible side chains. Now you can have these possible side chains attached to your porphyrin ring. Let's see how. And depending upon what side chains are attached and how they are attached, you're going to end up with different types of porphyrins. Okay. Okay. So you have based on the side chains, three types of porphyrins. And what are they? That is UCP. Okay, now what is UCP? Your U stands for Europorphyrin. Europorphyrin. Okay, C stands for coproporphyrin. And P stands for protoporphyrin. Okay, now let's see what is the difference between these three. So in Europorphyrin, I'm representing the pyrrole rings like this simply. The correct structure I've already represented. This is just for representation purposes only, okay? Not drawn to accuracy. So just imagine this is your porphyrin ring. So I am going to draw the structure of uroporphyrin. Uroporphyrin. Okay, uroporphyrin. Okay, I'm going to explain these numbers as well. What do you mean by 3 and 1? For now, so for your uroporphyrin, you have A and P, that is acetyl and propionyl. Then again, you have A and P, that is acetyl and propionyl. Then you have A and P, that is acetyl and propionyl. And then you have P and A. So you see, the pattern here is reversed. Reversed. If it was A and P, they were alternating. So it would have been uroporphyrin 1. Okay, in uroporphyrin 1, your acetate and your propionate, they are alternate. So in uroporphyrin 3, it is reversed. And in uroporphyrin 1, your acetate and propionate, they alternate. That is, if I draw the structure of uroporphyrin 1, it is going to be like this. AP, 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 it's going to be AP. So this is your uroporphyrin 1. Okay. I hope it's clear. So this was your uroporphyrin 3, where the A and P here, they are reversed on the ring 4. Whereas in uroporphyrin 1, your A and P, that is acetate and propionate, they are alternating. Okay. So that was your uroporphyrin. Now talking about coproporphyrin. In coproporphyrin, the side chains are M and P. What is M and P? That is your methyl and propionate. So if I draw coproporphyrin, it is going to look like so. In coproporphyrin, you have M and P, M and P, M and P, and M. Again, this is coproporphyrin 3, okay? Because in humans, we are only having the type 3 isomer, okay? So therefore, again, you can see this is reversed. Now, in protoporphyrin, what is happening? So in protoporphyrin, the first and the second propionate in your coproporphyrin is changed to vinyl, okay? So that is, this is the first and this is the second propionate. So this is changed to vinyl here. So in protoporphyrin, you have M and V, then you have M and V, then you have M, P, and P and M. So you see your coproporphyrin and protoporphyrin, what is changing? Only the first and second propionate changed to vinyl, okay? And in coproporphyrin, you have P and M And in uroporphyrin, you have A and P as the side chains. Okay, so that was about the different types of porphyrins. Now, one more thing is left, that you have different isomers of porphyrins. Okay, so porphyrins have different isomers. Porphyrins have different isomers. Okay, many different isomers are possible for porphyrin, out of which two are important. Okay, that is type 3 isomer and type 1 isomer. So your type 1 isomer, it is not normally present in our body, not normally present in our body. So in type 1, we've already seen that your side chains are alternating, and in type 3, they are present in our body. And in type 3, we have seen how the side chain was reversed on the fourth ring. Okay, and if what is happening, your type 3 is formed enzymatically. Okay, the enzyme is going to act and form type 3. If enzymatically your type 3 isomer is not being formed, then only in that case your type 1 isomer is formed. Otherwise, type 1 isomer is not normally present in our body. Okay, so with this, I've explained you all these structures. Now moving forward to heme synthesis. So let's quickly recapitulate all that we've studied. Firstly, talking about the structure of heme, that it is a ferroprotoporphyrin. I explained what is ferroprotoporphyrin, that it is having iron in the center of the porphyrin ring. What is a porphyrin ring? It is a 4-tetrapyrrole ring attached by methylene bridges. Okay, so this is your porphyrin ring. Okay, and to the center, when you have iron coordinated, then it becomes heme. Okay. Now I said that your porphyrins, they are of three types based on the possible side chains. And what are the possible side chains? They are methyl, propionyl, vinyl, and acetyl. So based on these possible side chains, your porphyrin is of three types. And what are those three types of porphyrins? That is uroporphyrin, coproporphyrin, and protoporphyrin. So in uroporphyrin, you are having the side chain acetyl and propionyl. And in coproporphyrin, you having the side chains methyl and propionyl, and in protoporphyrin, the first and the second propionyl of coproporphyrin is changed into vinyl. Okay, so that was about the different types of porphyrins. So porphyrins are having different isomers, out of which two isomeric forms are important, type 1 and type 3. So your type 1 is not normally occurring in the body. It is synthesized only when enzymatically your type 3 isomer is not synthesized, whereas your type 3 isomer, they are present in the body. And your protoporphyrin 9 belongs to the type 3 isomer series. Okay, now before moving forward, so we have seen the structure of heme. Now before moving forward, let me quickly give you a list of all the proteins in a body that contain hemes. So heme containing proteins in our body. So first one, very important, is our hemoglobin, followed by myoglobin. Then you have cytochrome P450. Then you have nitric oxide synthase, that is NOS, nitric oxide synthase. Then you have catalase. Then you also have tryptophan pyrrolase, tryptophan pyrrolase. Okay, so these are the list of some proteins in our body that are having heme in its structure. And the structure of heme, I hope it's very clear. Now let's move forward to synthesis of heme. So talking about heme synthesis, firstly, if I take the site of heme synthesis, so where is it occurring? Site. So it is taking place in almost all the tissues, synthesized in almost all tissues. Okay, but predominantly in, it is synthesized in liver and erythroid precursor of bone marrow. Also, there is an exception that where your heme is not synthesized. So, it is not synthesized in mature RBCs. So always remember, heme is not synthesized in mature RBCs. And why is it not synthesized in mature RBCs? Because our mature RBCs, they lack mitochondria. And the initial and the last three steps of heme synthesis, they are taking place in the mitochondria. So talking about the organelle, so it is both cytoplasmic and a mitochondrial cycle, because the initial and last three steps are taking place in mitochondria. Okay, so that was about the site and the organelle of heme synthesis. Now talking about the steps of heme synthesis. So it can be broadly classified into three major steps. Number one, it is the synthesis of porphobilinogen. So formation of porphobilinogen. This is a monopyrrole. That is only one pyrrole, that is porphobilinogen. This is followed by synthesis of porphyrin, synthesis of porphyrin, and then chelation of iron. So these are the three major steps. Okay. Now let's begin with the detailed individual steps of heme synthesis. So the first step, this is taking place in the mitochondria with the help of the mitochondrial enzyme ALA synthase 1. Okay, so ALA synthase 1 is bringing about the first step. And what is happening in the first step? Your glycine combines with succinyl CoA to form delta amino levulinic acid. That is ALA. Okay, so in the first step, what is happening? Your ALA is being synthesized and PLP is acting as the coenzyme required. Okay, so this step, this is the rate limiting step. Rate limiting step. PLP is required, and this is inhibited by heme. Okay, and this ALA synthase, it has two isoforms. Okay, that is ALA synthase 1, which is used here in this reaction, as you can see. ALA synthase 1 and ALA synthase 2. So this enzyme is having two isoforms. So your ALA synthase 1 is present in all the organs, especially liver. Present in all organs, especially liver. Whereas your ALA synthase 2, it is present in erythroid precursors. Present in erythroid precursors. Okay, so the synthesis is based on availability of globin. Okay, so here, since this is occurring in precoid synthesis, so the synthesis of heme is going to be based on the availability of globin, because the heme here synthesized is probably going to combine with globin to form hemoglobin. Okay, so depending upon the presence of globin, if there is more globin, so there should be more heme. Heme synthesis is going to be increased to form more hemoglobin. So it, as it is seen in erythroid precursors, it is most probably that this heme will be combining with globin to form hemoglobin. So more the globin, more is going to be the heme formation. So more globin, more heme synthesis. Okay, and the other one, that is ALA synthase, which is present almost all the organs. This is an inducible enzyme. enzyme, and this is induced by heme. Okay, so decreased heme. If the level of heme in the body is low, there is decreased heme. It is going to induce synthase 1. Okay, so the expression of synthase 1 is going to be synthase 1 expression increases, and this is taking place by induction repression mechanism. Okay, so that was about the first step of your heme synthesis, that is your glycine. It combines with succinyl CoA. This is taking place in the mitochondria. The enzyme is synthase 1. PLP is required, and you can see ALA being formed. Okay, now this ALA synthase, this enzyme is having two isoforms, that is ALA synthase 1 and ALA synthase 2. ALA synthase 1 is present in all the organs, especially in the liver. It is an inducible enzyme. If heme levels are low in the body, this enzyme is going to be induced, that is, expression is going to increase. And your synthase 2, this is present in erythroid precursor cells. So the heme that is going to be synthesized in the erythroid precursors by synthase 2, it is most probably going to be used for hemoglobin synthesis. It is going to combine with globin. Okay, so the expression of this enzyme or the synthesis of heme by synthase 2 is based on the availability of globin. So if there is more globin, there is going to be more heme synthesized. Okay, now talking about the next step, what is going to happen? Your synthase, two molecules of synthase, they are going to condense or join to form porphobilinogen. Okay, and this step is brought about by the enzyme dehydratase. Okay, so this is brought about by the enzyme dehydratase, and this is taking place in the cytosol. So your ALA that is formed in the mitochondria, it enters the cytosol. There it is acted upon by the enzyme dehydratase. And what is going to form? Two molecules of ALA, they are going to condense or join to form porphobilinogen. Okay, so your PBG is going to be formed. This is the short form of porphobilinogen, and this is a monopyrrole. Okay, so your monopyrrole is formed, and since this is a dehydratase enzyme, so water molecule is liberated or water is removed. Now, what happens? Your porphobilinogen, it is acted upon by another enzyme known as hydroxymethyl bilane synthase. Okay, so your four molecules of porphobilinogen, they are going to join to form hydroxymethyl bilane, represented as, I'm going to write this as HMG. Okay, and what is going to happen? They are going to join together, and your HMG is going to be formed. Now, this enzyme, hydroxymethyl bilane synthase, it has different names. So, this enzyme is having three different names. One is your HMG, that is hydroxymethyl bilane, HMB, sorry, synthase. Then, as you can see, ammonia is being released. Okay, so it is also known as PBG deaminase. PBG, that is porphobilinogen deaminase, and it is also known as uroporphyrinogen 1 synthase. Now, this HMB, that is hydroxymethyl bilane, that is formed. This is a linear tetrapyrrole. So what happened? Your glycine and succinyl CoA, they combined to form your ALA. Then two molecules of ALA, they combine to form your porphobilinogen. Then your four porphobilinogen molecules, they are going to combine. Porphobilinogen, as I said, was a monopyrrole. Okay. Now, four porphobilinogen, they're going to combine to form a linear tetrapyrrole, which is known as hydroxymethyl bilane. Okay. And the enzyme involved is known by three different names. It is known as HMB synthase, It is also known as PBG deaminase, and it is also known as, so HMB synthase, because of HMB is being synthesized. Okay, PBG deaminase, because as you can see, 4 molecules of ammonia are also being released. Now you might be thinking that why it is known as uroporphyrin 1 synthase. Okay, because one, as I mentioned, it is not usually formed in the body. So because I'll tell you why. Because this HMB, that is your hydroxymethyl bilane, it is going to have two fates. Okay, because this HMB, that is hydroxymethyl bilane, that is synthesized, has two fates. Okay, one fate, which is the predominant fate, is that it is going to be acted upon by the next enzyme, that is uroporphyrin 3 synthase. Okay, so this is the predominant normal route. Predominant normal route. And next is, what if this enzyme is absent? Okay, so the other route is that the enzyme is absent. That is your next enzyme, uroporphyrin 3 synthase is absent. Now this HMB, it is going to spontaneously cyclize to form, spontaneously cyclizes to form uroporphyrin 1. Okay, so what is formed? Uroporphyrin 1 is formed, and this uroporphyrin 1, as I said, that your isomer of type 1 kind, they are not normally present in the body. You do not have further enzymes to metabolize this. So because as there are no enzymes in the body to act on this type 1 form, further metabolism of this UPG1 is not going to take place. Okay, not normally present. Okay, no enzymes further to metabolize it. But this is only taking place when the next enzyme in the heme synthesis is not absent, which is not true, because it is usually there. So the predominant route of this HMB that is synthesized is that it is going to be acted upon by the uroporphyrin 3 synthase. And what is going to happen? The ring is closed. Your linear tetrapyrrole is going to cyclize and isomerization is going to take place, and you end up with uroporphyrin 3. Okay, so what is happening here? Your HMB cyclizes to form the first porphyrin. Okay, so as you can see, so HMB, it cyclizes to form the first porphyrin, which is your uroporphyrin 3. Okay. Now what is going to happen? Your next enzyme, that is UPG3 decarboxylase, is going to act, and you end up with coproporphyrinogen 3. Okay, this is a decarboxylation reaction. Why I say decarboxylation reaction? As you can see here, what is the difference between uroporphyrinogen and coproporphyrinogen? There is only the difference of your acetyl changes to methyl. As you can see here, your uroporphyrinogen, they are having acetyl groups which are changing into methyl. See, acetyl. So acetyl of uroporphyrin is changing into methyl. So your acetyl is having 2 carbons, as you all know, and methyl has only 1 carbon. So 1 carbon is lost and it is removed as carbon dioxide. So you have 4 acetyls, as you can see here, which are changing into methyl, 4 methyls. Therefore, 4 carbon dioxide, they are being lost. Okay. So now what are you ending up with? You end up with coproporphyrinogen 3. Now this coproporphyrinogen 3, it is going to change into protoporphyrinogen 9. Okay. Now what is going to happen? Here your 2 propenyl. See, this propenyl, It's going to change into vinyl. So your propionyl of coproporphyrinogen 3, it is going to change into vinyl. This one and here this. Okay, so you end up with protoporphyrinogen 3. So this is a decarboxylation and oxidation is taking place, and the enzyme is coproporphyrinogen 3 oxidase. This is a mitochondrial enzyme, as I had mentioned earlier also, that the last three steps are taking place in the mitochondria. Now what happens? Your protoporphyrinogen 9 is going to change into protoporphyrin, okay? Because the heme is having protoporphyrin and not protoporphyrinogen. Now, what is the difference between protoporphyrinogen and protoporphyrin? It is the difference is only between the bridges that are joining the pyrrole rings. As you can see in protoporphyrinogen, you have which bridges? You have methylene bridges. Okay, so here you have methylene bridge. Whereas in protoporphyrin, you have methylene bridges. Okay, so that is the difference. Your protoporphyrinogen 9 is going to change into protoporphyrin 9. Okay, and this 9, this belongs to the type 3 isomers. Okay, now what is going to happen? Your protoporphyrin 9 to this, and iron is chelated. As you can see, it is coordinated in the center here, and you end up with heme. And this ferrochelatase enzyme, this is a mitochondrial enzyme and it is inhibited by lead. So it is sensitive to lead poisoning. Okay. So that was about the steps of heme synthesis. I'll quickly recapitulate before we move forward. So you see how just glycine was combining with succinyl CoA to form ALA. Then that ALA, two molecules of ALA, they were condensing to form porphobilinogen. Then and this porphobilinogen, it was a monopyrrole. And in heme, we need tetrapyrrole. Now this porphobilinogen, they're going to combine to form a linear tetrapyrrole at the moment, that is hydroxymethylene bilane. Okay, now this hydroxymethylene bilane, it is going to cyclize, it is going to form uroporphyrinogen. Okay, now this uroporphyrinogen is having acetyl in its side chain, so that acetyl, it is going to be converted into methyl. Okay, so 4 carbons are going to be removed, so therefore you can see 4 carbon dioxide removed, decarboxyling taking place, and you end up with 4 coproporphyrinogen 3. Now this coproporphyrinogen 3 is again, its propionyl side chains are going to change into vinyl, and you end up with protoporphyrinogen 9. Okay, so now protoporphyrinogen is going to change into protoporphyrin. The difference only between the methylene, between the bridges, your methylene bridge is going to change into methanol bridge. And now in the last step, what is going to happen? Your protoporphyrin 9, it is going to be chelated with iron. Okay, now this step is spontaneous, but the rate is enhanced or increased with the help of the enzyme ferrochilase. Okay, so that was about the synthesis. Now talking about the factors affecting heme synthesis or regulation of heme synthesis. Okay, factors or regulation. Okay, firstly, as we have seen, what is going to affect is lead. Okay, so your lead, it was affecting 2 enzymes. What were those? The 2 enzymes that are affected are ALA dehydratase and ferrochilase. These are the 2 enzymes, ALA dehydratase and ferrochilase. And your heme was also involved in regulation. Your heme, it was regulating which enzyme? It was regulating ALA synthase. Okay, so when heme levels are low in the body, your ALA synthase expression is increased. That is the gene for ALA synthase, it is transcribed more. Okay, so the gene is transcribed more, and then you have certain drugs like barbiturates, They also enhance your heme synthesis. Okay. So drugs like barbiturates, they also induce heme synthesis, or you can also say, in other words, they aggravate porphyria. Let's see how. Because your barbiturates, they are metabolized by cytochrome. Okay. So your barbiturates, they are metabolized by cytochromes. That means your cytochrome is going to decrease. Okay. Now, cytochromes, they are containing heme. So therefore, what happens? Your heme also decreases, and decreased heme means that your ALA synthase is going to be increased. Okay, so your ALA synthase expression is going to increase, and therefore it is going to aggravate porphyria. Okay, so that was about the regulation of heme synthesis, or factors affecting heme synthesis. Okay, now moving forward to different types of porphyrias. Okay, that is the disorders associated with heme synthesis. So what are porphyrias? They are a group of disorders associated with deficiency or defect in any of the enzymes of heme synthesis. So if any enzyme in the heme synthesis pathway, it is defective or deficient, it is going to lead to a particular type of porphyria. Okay, and because of which various intermediates that we've seen in the heme synthesis cycle, they are going to accumulate. Okay, so the intermediates of heme synthesis, they are going to accumulate. They accumulate. Now, most of your porphyrias, almost all porphyrias are autosomal dominant, with the exception of, so with the exception that I'm mentioning below, rest of all your porphyrias, they are autosomal dominant. So your X-linked protoporphyria, that is one exception, followed by congenital erythropoietic porphyria, congenital poietic porphyria. Then you have ALA dehydratase deficient porphyria. And then you have erythropoietic porphyria. Okay, so with the exception of these, all your porphyrias, they are autosomal dominant. So your autosomal dominant, they manifest in the late ages, and your autosomal recessive, they manifest in young ages. Okay, now let's have a look at the different types of porphyrias. So here is this chart which has all the porphyrias summarized together. As you can see, this is the mitochondria. In the mitochondria, the first step is taking place. And the first step, as we all know, it is controlled by heme. Okay. So if there is excess heme, this first enzyme, it is going to be inhibited. Now this ALA, that is delta aminolinoleic acid, that is formed, it enters the cytoplasm. As you can see, in the cytoplasm, it is forming porphobilinogen. Okay, and the enzyme was ALA dehydratase, which was actually inhibited by lead. Okay, so this enzyme ALA dehydratase, it is inhibited by lead, and lead is also inhibiting your ferrochelatase. Okay, that was changing your protoporphyrin 9 into heme. Okay, so your ferrochelatase and ALA dehydratase are particularly sensitive to inhibition by lead. Your protoporphyrin in ALA accumulates in the urine, and ALA dehydratase deficiency porphyria, it is a very rare autosomal recessive acute hepatic porphyria. Okay. And another point I'd like to add to this is and it is also known as Porphyria, since it resembles lead poisoning. Okay. Now what happens? Your porphobilinogen, it changes into hydroxymethylbilane. Then this hydroxymethylbilane is changing into uroporphyrogen 3. Okay. So this changing of porphobilinogen into hydroxymethylbilane, the enzyme is your hydroxymethylbilane synthase, and when this enzyme is deficient, then you end up with acute intermittent porphyria. Okay. So this is an autosomal dominant disease, and your porphobilinogen and ALA accumulate in the urine. The urine darkens on exposure to the light. So when the urine is voided, then it is colorless, but on exposure, it is going to darken. The patients are not photosensitive. Okay. So urine, when voided, is colorless, but on standing, it is going to darken. Now, next, when your enzyme uroporphyrogen 3 synthase, which is forming uroporphyrogen 3, here, this is defective, you end up with congenital erythropoietic porphyria. Okay. Congenital erythropoietic porphyria. This is an autosomal recessive disease, and it is caused by uroporphyrogen 3 synthase being deficient. Okay. Now here your uroporphyrogen 1 and porphyrogen 1, they are going to accumulate in the urine, and patients are photosensitive. Now, this is important that here the patients are photosensitive, and major manifestations are related to skin. So, since the patients are photosensitive, therefore, major manifestations related to skin. And another unique thing about this is monkey face. What is monkey face? Due to repeated attacks of dermatitis and scarring, it is going to lead to a typical face, which is referred to as monkey face. Okay. So that was about congenital erythropoietic porphyria. Then moving on to the next enzyme, which is your uroporphyrogen decarboxylase. If that is defective, you end up with an autosomal dominant disorder, that is Porphyria cutanea tarda. Okay, here your uroporphyrin is going to accumulate in the urine. It is the most common porphyria, and it is photosensitive. Okay, and it is photosensitive with blistering photosensitivity. Okay, so blisters are going to be formed. So blistering photosensitivity, and also this is the most common porphyria, but here 80% of the cases, they are sporadic, and 20% are because of genetic mutations. Usually if you are having any enzyme deficiency, it is going to be because of genetic mutations. But here in this case, that is in Porphyria cutanea tarda, 80% of the cases, they are sporadic. That is, no genetic mutation is there. Therefore, it is also readily treatable. Okay. These are because of certain inhibitors for this enzyme. Okay. So your enzyme, that is your uroporphyrinogen decarboxylase, it is inhibited by certain substances like your iron. Okay, so 80% of the cases, they are sporadic. There is no genetic mutation. Therefore, it is readily treatable. And there are inhibitors for this enzyme, important inhibitor being your iron. Okay. Now, this coproporphyrinogen, it is going to move into the mitochondria, and the next 3 steps, that is the last 3 steps of the synthesis, they are going to take place in the mitochondria. So if your next enzyme, that is porphyrinogen 3 oxidase, that is deficient, you are going to end up with autosomal dominant hereditary coproporphyria. Okay, and there here what is going to accumulate your coproporphyrinogen 3 and other intermediates prior to the block, they're going to accumulate in the urine, and patients are photosensitive. Now moving on to the next enzyme deficiency, that is your protoporphyrinogen oxidase. If that is deficient, you have an autosomal dominant disease, that is variegate porphyria. Here, what is going to accumulate? Your protoporphyrinogen 9 and other intermediates prior to the block, they're going to accumulate, and patients here again, they are photosensitive. Now, moving on to the last one, that is due to deficiency of ferrochelatase, you end up with erythropoietic protoporphyria. Here, what is going to happen? Your protoporphyrin accumulates in erythrocytes, bone marrows, and plasma, and patients are photosensitive. But this is non-blistering photosensitivity. Okay, so with this, we come to the end of all the porphyrias. Okay, and one important concept that you should know, that when you're going to have photosensitivity and not, so concept of clinical feature, that is those enzymes which are coming early in the pathway. Okay, so early enzyme deficiency, if those enzymes are deficient, that are coming earlier, then what is going to accumulate? Your ALA and your PBG, that is porphobilinogen, they are going to accumulate, and there will be no photosensitivity. But those enzymes which are coming later in the cycle, okay? So later enzymes, they are deficient, then what is going to accumulate? Your porphyrins are going to accumulate. So your porphyrins are going to accumulate, that is after PPG deaminase. Okay, if the deficiency is after PPG deaminase, other later enzymes, then your porphyrin is going to accumulate, and photosensitivity is seen. Okay, so with this, we come to the end of porphyrias. Okay, now let's talk about heme degradation. So your senescent, old, injured RBCs, or after they've completed their 120 days of lifespan, okay, in the circulation, then they are going to be phagocytosed, or other injured RBCs, they are phagocytosed by macrophages, particularly in the liver and the spleen. Okay, now what is going to happen? This is your old RBC. So we're done with heme synthesis, we're done with porphyrias, now we're talking about heme degradation. Okay, so I'm saying that old RBCs or injured RBCs, they are phagocytosed by macrophages. Okay, now what happens? Your RBCs, they are having hemoglobin, so hemoglobin is liberated from the RBCs. Your hemoglobin comes out, and hemoglobin, we all know, it is composed of heme and globin. So heme and globin, they are separated, they are split. So your globin, and it enters the amino acid pool in the body. Whereas your heme, it is acted upon by heme oxygenase to form biliverdin, and what is liberated? Your iron is liberated. This iron that is liberated, it is recycled. We've already studied the recycling of this iron in when we were talking about mineral metabolism in iron chapter. Okay, so now this heme is acted upon by heme oxygenase, your iron is removed, biliverdin is formed. This biliverdin is then reduced. Okay, how is it reduced? By the action of the enzyme biliverdin reductase, and what is formed? Bilirubin is formed. Okay, so your biliverdin, it is converted into bilirubin. Your biliverdin, it is a green pigment, and your bilirubin is a red orange pigment. Okay, I'll repeat this again. What is happening? Your macrophages, they're going to phagocytose the senescent RBCs, and then your hemoglobin is going to be liberated from the RBCs. And hemoglobin, it is split into globin and heme. The globin is going to join the amino acid pool in the body. The heme is acted upon by an enzyme known as heme oxygenase, and your iron, which is removed from the heme, it is going to be recycled, and it is going to be a source of iron in the body. So, and this is the major source of iron in the body, okay, and the other source of iron is through diet, okay. And then your biliverdin is formed. This is linear in structure, okay. And then this biliverdin, which is a green pigment, this is acted upon by another enzyme, biliverdin reductase. So biliverdin is reduced to bilirubin. Now this bilirubin, it is going to be transported to the liver via the blood, complexed with albumin. Okay, so bilirubin, albumin, we've already studied when we were talking about plasma proteins yesterday, that your albumin, one of the major functions of albumin was to transport ligands. Okay, so it is going to transport your bilirubin in the blood to the liver. Okay, so your bilirubin albumin complex, it reaches the liver. Here, this bilirubin, it is taken up by the liver by facilitated diffusion, taken up by liver by facilitated diffusion. Okay. Now in the liver, what is going to happen? Conjugation is going to take place. This is taking place in the liver cell. So your bilirubin, it is conjugated with glucuronic acid. Okay. So two molecules of UDP glucuronic acid are going to conjugate to bilirubin, and the enzyme is bilirubin UDP glucuronosyl transferase. Okay, and you end up with bilirubin diglucuronide. Okay, so in the first step, it is a two-step of conjugation. Bilirubin monoglucuronide is formed, then again another glucuronic acid is being added, and ultimately you end up with conjugated bilirubin, which is bilirubin diglucuronide. Okay, and then this bilirubin diglucuronide, it is going to enter bile through the common bile duct. Okay, so here this is another diagram so that you're able to understand. This is just a repetition of what I've told you. See, your dead and senescent RBCs, they are broken down. They are phagocytosed by the macrophages. Okay, and then I've explained how your heme is being changed into bilirubin. Then that bilirubin, it is taken up by the liver, and then what happens in the liver? It is conjugated with glucuronic acid. Then after conjugation, it was secreted into the bile, the common bile duct, and reaches the intestine. You can see here it has reached the intestine. Now what happens? In the intestine, your deconjugation is taking place. Glucuronic acid is removed via a bacteria, your intestinal bacteria, and the resulting bilirubin is converted into urobilinogen. Okay. So deconjugation is going to take place in the intestine by the bacterial flora of the intestine. So the bacterial enzyme beta glucuronidase is going to act, and your bilirubin is unconjugated. Okay, now what is going to happen? It is converted into urobilinogen, and this urobilinogen, it is colorless. Okay, now let's see what is happening. This unconjugated bilirubin that you have, 80% of this, it is excreted in the feces. Okay, so this bilirubin, unconjugated bilirubin, 80% of this, it is excreted in the feces. So urobilinogen is oxidized by the intestinal bacteria to brown stercobilinogen, and this stercobilinogen, it gives the brown color of the feces. Okay, now the remaining 20%, it is reabsorbed from the gut. Okay, remaining 20% is reabsorbed from gut. Okay, so out of which partly a portion of the urobilinogen, it participates in hepatic circulation, okay, in the enterohepatic urobilinogen cycle. So from the liver, which is again entering the bile, okay, and partly what is happening, the remainder of the urobilinogen, it is transported by the blood to the kidney, where it is converted to yellow urobilin and excreted, giving the urine its characteristic color. Okay, so part of it is excreted in the urine, part of it, 80% of it as urobilinogen, is changed into stercobilinogen and excreted in the feces, and part of it is actually entering enterohepatic circulation. Okay, so that was about the excretion of bilirubin from our bodies. Okay, and then if there is increased or elevated levels of bilirubin in the body, we end up with hyperbilirubinemias. Okay, so this we are going to discuss in the next class. So we've seen how heme is degraded, it is converted into bilirubin, how it is excreted. Now we're only left with the clinical portion, that due to certain reasons, sometimes your bilirubin levels may be elevated in the body. So we're going to talk about this the next class quickly before we begin with our next class topic. Okay. So I hope the lecture was useful. We covered a major chunk, only a small portion is left. So if you have any queries, you can ask before we end the slide. Okay, thank you for watching. See you in the next class.